temática es clones resistentes obtenidos por métodos biotecnológicos. El expositor, el doctor James Dahl, profesor investigador de la Queensland University of Technology. Adelante y bienvenido.
So good morning from Australia. There seemed to be a little glitch somewhere between Ecuador and Australia a little while ago, but now I think we're live. So thank you for the invitation to come and address your conference today. I'm going to be talking about resistant clones obtained by biotechnological methods, and that's both genetic modification and gene editing. So I'm going to cover both of those over the next period. So Australia, what's TR4 like in Australia? It's been here since around about 1990. It's endemic in the Northern Territory. The Northern Territory actually was a very, very small producer of bananas. But there's only one plantation or commercial plantation left in the Northern Territory in Australia, and that's the one on the top left of your screen. It's also the plantation where we have our field trials. TR4 also has very limited distribution in North Queensland. That's where the major production area of bananas in Australia is. It's on only a few plantations, and there's a huge effort to control its movement, and that seems to be, to a certain extent, successful. We are getting increasing numbers of plants infected, but those numbers are very small. So the strategy of reducing spread is working quite well, but is not eliminating spread. So when we look at how we can genetically improve plants, and so this is all plants, it really doesn't matter. On the top there's hybridization, that's the normal conventional breeding process. There are natural mutations, and there's also mutagenesis. So you would say under natural mutations, the GCTCV218 would fall into that category, and mutagenesis would be, for instance, the Rahan meristem resistant clones, which have been mutagenized. Then you have a group of technologies known as gene editing, SDN1, and SDN2, and SDN3, and they differ in their definitions. And then finally down the bottom there we've got transgenics and cisgenics. So they're basically the way that we can genetically improve plants. When we bring that down to Panama disease and bananas and TR4, essentially we've got four options. One is tolerant selections, and we've just heard two presentations regarding tolerant selections. There's conventional breeding, and certainly that is quite well advanced now in a number of areas, and that will result in new cultivars. It won't result in improved Cavendish, it'll result in new cultivars. And then we have genetic modification and gene editing, and those are two areas that I'm going to be talking about. So essentially, if we use conventional breeding, we have to develop a new banana cultivar that is different to Cavendish, and that's primarily because we can't introgress genes into Cavendish. So we're going to have a different cultivar, but it will have to have disease resistances. It will have to be high yielding, and it'll have to be transportable and have excellent taste and texture, and that's what's been selected for now. On the other hand, genetically modified or gene edited Cavendish, we're starting with Cavendish, and so what we're doing is adding multiple disease resistances. So it's just a different strategy altogether. So KUT's TR4 R&D program, we've been working on TR4 for a long time. We started our research way back over 21 years ago when we started looking in wild bananas for TR4 resistance genes. And then we were able to isolate a gene that we called RGA2. It's a resistance gene from a wild seeded banana called Musáceas malacensis, which occurs quite widely through Southeast Asia. And that's probably where TR4 evolved, and that's very common that you find resistances where the disease and the plant both co-evolved, and that's certainly in Southeast Asia. So in 2006, we started the genetic modification of Cavendish, and we put in 2 genes. One was a gene called SED9, which is an anti-apoptosis gene, and the other one was this RGA2 gene, which we'd isolated from bananas. And we also looked at resistances in one of our local cultivars, ladyfinger. We developed race 1 resistance to ladyfinger. That was in the glass house. Most importantly, we transferred the Cavendish plants that have been genetically modified with SED9 and RGA2 into the field in 2015. And then we began our gene editing program for Cavendish in 2017. That's for Tr4 resistance. And then we also went into phase 2 of our Tr4 resistance field trial in Darwin, a much bigger one, which I'll talk about. And this year, we're now preparing the dossier for the deregulation of what we call QCAP4. It's one of our GM lines, but we've also now generated non-GM gene-edited Cavendish. So both of those are pretty significant opportunities for us. So firstly, the GM program, and that importantly was to identify that resistance gene. And this is Musáceas malacensis on the left-hand side. It's a seeded diploid, which occurs in the wild. This particular accession was one that we were able to access, and we knew that there was a single dominant Tr4 resistance gene in that. And so we were able to then go and look for what that resistance gene was. And we started, as I said, in 2001. And through various technologies, genome analysis, expression in resistant versus susceptible siblings, etc., we were able to identify a very, very promising gene. And we transferred that into Cavendish by genetic modification. So there's a banana gene going from one banana to another. And we did our first field trial in Darwin starting in 2015. So I've just diagrammatically there demonstrated that we've taken a single resistance gene, RGA2, from Musáceas malacensis and transferred that into Cavendish, and the one we used was Grand Name, probably the most popular of all of the cultivars of Cavendish. We started our field trials in 2012, the first field trials, and on this slide, that's a map of Australia. You'll see on the middle right-hand side, that's where we work at QUT, Queensland University of Technology. We're in the subtropics. Our field trial, and we're in Queensland. And I've also put in there the Tully Valley, and that's where the major banana producing areas are in Queensland. The field trials were at a place called Lambles Lagoon, which is up in the Northern Territory. It's quite an isolated location, and we're working on the last commercial plantation in the Northern Territory. So this field trial, it's on a large commercial plantation which has been severely affected by Tr4 for more than 10 years. And the field trial site was adjacent to commercial plantings. Very importantly, we've got a complete history of every plant in the trial, and we confirm the infections by PCR. PCR has become very famous over the last 2 years because it's the technology also used to detect COVID-19. So we collected invaluable data, and this is some of the really important data that we'll be publishing soon on rates of infection, spread, recovery, etc., and specimen collection. So that's the field trial site you see there. So we've got a field trial site. We've also got screen houses where we're now developing as early stage screening before plants go into the field. And we've also got a small, what we call a field lab. It's a lab where we can do DNA extractions and we can grow up the fungus so that we can inoculate plants in the screen house. Okay, so that trial was planted in 2012. It ran for 3 years. we had 4 controls, GCTCV218, which is also formosana. You've just heard about that. Also another mutant called DPM25, which is supposed to have resistance to subtropical race 4, and also grandnane and Williams. So both of those Cavendishes were part of the trial. Okay, this is the outcome after 3 years. And in the blue bars, they're the controls. And as you will see, after 3 years, DPM25, which was apparently resistant to subtropical race 4, had no resistance to TR4. 100% of the plants after 3 years were infected. Then grandnane, and then formosana, GCTCV218, after 3 years, had about 68% infection. Williams was marginally better than that, probably about 66% infection. More importantly, on the red bars on that graph are the genetically modified lines, and one of those, RGA23, had no infected plants after 3 years. And 3 other lines, RGA22, 24, and 25, had very high levels of resistance. We had 2 lines in there which also didn't have any particular resistance to TR4. So 4 of the 6 lines on that graph had high levels of resistance, and 1 was close to immunity. And that just gives you an idea, this graph, of the rate of infection over those 3 years. And so in the red lines are the controls, and the green lines are the genetically modified lines. And so you'll see that we've got lines right down the bottom there, just tracking along the bottom, at virtually no plants infected after 3 years. So that was, and this is just slides. This is on the final day of the trial, and you'll see on the left-hand side the non-GM controls, how very severely affected they were, and then our RGA23 line, which after 3 years still looked excellent in the field. And we've got further plants of those on the right-hand side. One of the most important things, and this is well, it may be a little bit technical, this is demonstrating that the RGA2 gene not only occurs in the genetically modified lines, but it actually occurs in Cavendish as well. And we now know that that gene is present in virtually every banana. So why doesn't it work? Why is grandnane, which contains the RGA2 gene, still susceptible? And the reason for that is shown on this graph. And this is what's known as a messenger RNA expression, mRNA, as everybody knows about mRNA from COVID-19. So this is messenger RNA, mRNA, and you'll see on the far left-hand side there that grandnane has very, very low expression levels of RGA2, of the RGA2 gene. And then you'll see the 4 resistant lines in the middle there, and they all have relatively high levels of expression. So the reason that grandnane is susceptible and that the RGA2, 2, 3, 2, 4, 2, 5 are resistant is that that gene is essentially turned off in grandnane, and that becomes more important as we go along. We've published that. We published that in 2017. One of the things we are very keen to do is to publish our work so that everybody has access to it in highly reputable journals. We then went into our expanded field trials, and these were to take the best of these plants through to deregulation. So the expanded field trials, these contained large numbers of the RGA2 lines that you just saw in the previous field trial. So that was a small field trial, the initial one. This is a much, much larger field trial. And so we get a much better idea, particularly of things like cycle time and bunch weight. We also have new RGA2 lines in that field trial of both grandnane and Williams. So the originals were only in grandnane, so we've got new lines coming through in Williams as well. And these, we also have a number of lines coming through with different resistance genes, because Malacensis, or the line that we had, isn't the only wild banana that's resistant to Tr4, and so we're looking for additional resistance genes so we can start to stack those. This second field trial was planted in early 2018, and again, this second field trial was up in Darwin at Lambles Lagoon. So you can see from this slide. So that's the field trial from a distance in that top photo, and then in the middle there again a close up of the trial, and on the left and right hand side some of the bunches, particularly of the RGA2 lines. So this second field trial, as I say, is a much larger field trial, and it's going to be over 5 years. We've already gone for 3.5 years. It's a randomized block design. Very importantly, this trial has been designed to mimic a commercial plantation. So it's managed as if it's a commercial plantation. So, and we take all sorts of very important data. All the plants were transferred to Darwin Fruit Farm. That's where the field trial is, in vitro, and they were acclimatized there over 3 months. So there are 4 original grand name lines, and they're the only ones I'm going to report on today. The results were almost identical to the phase 1 field trial. But not only did we collect data on the infection rates, but we also collected data on yield, on cycle time, and very importantly, we had the long-term history of each plant. And we're just in the process of finalising a manuscript for the first 3 years of that trial. So what's happened over those first? I'll show you a couple of photos just to demonstrate again what Tr4 looks like in the Northern Territory. You'll see on the left-hand side there, these are control plants, and how we get that vascular necrosis, very severe vascular necrosis in the pseudostem of the control plants. All of the infections are determined or are confirmed by PCR. So we've now got very specific tests for Tr4, for subtropical race 4 and for race 1, and within race 1 the different BCGs. So what we do is we extract the DNA on site and then transport it to our labs in Brisbane, and we do PCR, but we also sequence the PCR product either directly or through cloning, and that's to just absolutely be certain that the infections that we're dealing with are caused by Tr4. Okay, these are the results of the second field trial, and this is after 12 months. So what you're seeing after 12 months is grand name. So remember, this is trying to mimic a commercial plantation. So we didn't add Tr4 to the field. We planted in a heavily infested field, but we didn't add more in inoculum into that field. So after 1 year, grand name 16%, and we now have new names for those lines. They're now called QCAV 2, 3, 4, 5, and QCAV4 after 1 year had no infected plants, and this is over a large number of plants. After 2.5 years, the infection rate in grand name had gone up to 52%, and you were starting to see little bits of infection in some of the GM lines. The QCAV4, for instance, and QCAV3 still had very, very high levels of resistance. It's after 2.5 years, not after a plant crop and a first return. Then we go to 3 years, and now we're starting to see much higher levels, and I've put Williams control in there as well. So after 3 years, we've got 68% infection in the Williams controls, these are non-GM, and 58% in the grand name. Again, QCAV4, no infected plants, and QCAV3 with just a very few plants infected. So both of those lines showing very, very good levels of resistance after 3 years. These are the results after 2.5 years. They're the only ones that we've fully analysed, and this is yield per hectare, and it equates to particularly bunch weight, of course. So after 2.5 years, if you compared the grand name controls, yield per hectare was round about 25 tonnes per hectare. QCAV4 was around about 54 tonnes per hectare. So pretty impressive yield from QCAV4. So that's what it looks like after 3 years. We're now out, so we've done, and here you'll see we've got the plant crop plus 5 ratoons, and we've now got 1 or 2 plants of QCAV4 just showing some infection, but you'll see the differences are spectacular there when we see Williams and Grand Nain after 6 cycles are getting up to 90% infection, and with the QCAV4 we've got round about 2% infection. That's after 6 cycles. And then we can have a look at, as a line graph as well, how that progressed over the different crops, from plant crop through ratoon 1, 2, 3, 4, and 5. So that's amazing data that we now have. So the very important outcomes from this phase 2 field trial is that after 6 crops, that's a plant crop plus 5 ratoons, we have 1 line which is QCAV4. So out of 300 harvests from that QCAV4 line, only 2 harvests have been lost to infection. So that's a minuscule amount. But most importantly, and we're going to publish this data, the bunch weight of QCAV4 is equivalent per plant to the grand name control. And amazingly, the cycle time is slightly shorter than the grand name control. And that was, we certainly didn't expect that. We expected it to be around about the same. So certainly this line, it contains a single banana gene, and this banana gene, RGA2, occurs in virtually all domesticated bananas. So the outcome, we've developed Cavendish bananas with close to immunity to Tr4, so they're now a reality, and that there is no impact on yield, either through bunch weight or through cycle time. So they're very, very important outcomes. Now I want to move on to the next generation of technologies, and that's gene editing, and we're doing gene editing in Cavendish, but also other bananas. So to give you an idea of what the difference between genetic modification, which includes transgenics and cisgenics, and gene editing. So essentially, genetic modification involves the addition of new DNA into the plant through transformation, and almost always includes what can be called new or foreign DNA. So in the case of QCAV4, that foreign or new DNA is the RGA2 gene, which comes from bananas. So it's not particularly foreign to bananas, but it is still new DNA. And because it's new DNA, additional DNA, they're classified as genetically modified. However, gene editing is quite different. Gene editing is very much the precise and targeted modification of a plant's DNA, and these can include things like deletion, substitutions, etc., or through homologous recombination of entire genes. So for non-GM gene editing, and that's why we're particularly interested in, there must be no new foreign DNA added. So what we're doing is just going in and manipulating the DNA, the genes that are already present in Cavendish bananas. So the components of that, we need a technology for editing bananas. We need the ability to transiently edit without any new DNA being incorporated. we need an editing strategy, and I'll talk about that. And we've got a donor genome and a recipient genome. So the editing technology we use is CRISPR. We have a license for CRISPR, so that allows us to do these experiments. And it's the technology that the two discoverers won the Nobel Prize last year for. And it allows us to go in and just edit very, 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 very precisely and very, very particular genes. Okay, so what we've done previously is demonstrated that this CRISPR technology works in bananas. And the way we did that was we took our embryogenic cell suspensions and we knocked out a gene called phytoN desaturase. Now, what phytoN desaturase does, it's part of the carotenoid pathway, but when you knock it out, you end up with albino plants, so plants that produce no chlorophyll. So it's a very good marker, if you like, of the ability to edit a plant. So here's some previous examples, so poplars, apples and petunias, and on the right-hand side, these albino plants of each of those. So bingo, we were able to do exactly the same thing in bananas. And what this showed was that we could go into the banana genome, we could switch off a gene, and we could visualize that, because that once that happened, that we ended up with albino plants. Clearly, this isn't a commercial trait. Bananas without chlorophyll will never be commercialized, obviously, but it provides us with a marker to be able to develop the technology to be able to do that. And so we could actually make bananas which were completely white, in which case all three alleles were knocked out. If we knock out 2 out of 3, we get a pale green plant, and if we only knock out 1, they look quite normal. So it's possible to manipulate how many of the copies of individual genes we knock out. And that's just to show that these characteristics are inherited from one generation to another, which is obviously very important. Now we've published that work, so that's already out there. We know that this is actually a very, very efficient process in Cavendish, so that gives us the opportunity now to go forward and do real edits. So the big challenge has been to develop a platform technology for developing edited Cavendish bananas that obviously have been edited, that do not contain, this is really important, do not contain any foreign or new DNA, and that can be adapted to many, many different banana cultivars. Then the next stage is identifying genes that can be either upregulated or downregulated to provide new traits such as Tr4 resistance. And we have to use edits that are acceptable in different countries. And the really important part about that is that in many countries, and I'll show you this at the end, the gene editing, the sort of gene editing that we're doing, is not considered genetic modification. So it doesn't require any regulation and it doesn't require any labelling. And the number of countries that are adopting this, particularly in North and South America, and obviously in Australia, Japan, and now more in Asia, are accepting non-GM gene editing. Okay, so where do we go? So firstly, we needed to be able to edit and regenerate plants. We had to confirm the edits by sequencing, and then we had to demonstrate, very importantly, that we could do this without any new DNA being incorporated. So we did this in 2 ways. One is by PCR, and then ultimately by sequencing the entire genome of those plants. And we use this phytoN desaturase gene as a convenient visual marker to start with. Right from the very beginning, we considered protoplasts. And protoplasts are single cells where the cell wall has been dissolved away So each one of those cells is very, very much an individual. Very important reasons for doing that, and that's so that we could edit those, and the edits when we regenerated a whole plant would have come from just one cell. So that's a very important part about that. And so we've been able to generate protoplasts from our embryogenic cell suspensions. That was fabulous. But also from the meristems of in vitro grown plants, and again, also from immature male inflorescences. So that's been a big breakthrough for us, to be able to generate these protoplasts, and at very, very high levels. You see those numbers on the right-hand side. They're up in the tens of millions, which is absolutely fabulous. We were then able to demonstrate that we could put DNA into these protoplasts, and that the DNA would work, as in the DNA would express the genes we put in. And in this case, we used plasma DNA, and we put in a gene that encodes a green fluorescent protein. And so you'll see when we do that, from the top to the bottom, we were able to show that we could get this DNA into these protoplasts, and it was expressed. So that was a very, very important breakthrough, and that's known as transfection. So then the big challenge, of course, was, could we regenerate whole bananas from protoplasts? And very, very little has been done on this in the past. So we've taken protoplasts, we've embedded them in agarose beads, and you'll see in the top photographs there, that top panel, these protoplasts form cell walls, as you'll see in that third photograph, the jars forming cell walls after 3 days, and then they start to divide. And the wonderful thing is, by manipulating the media that we grow them in, the food that we give them to eat, we can get them to go all the way through to somatic embryos, and that's the photo on the bottom left-hand side there. But not only that, we're now able to take those somatic embryos, we can germinate the embryos, and we can get whole Cavendish plants back. So in the bottom there, you'll see 4 individual bananas. They're Williams bananas, and they were generated from protoplasts, and that process took between 7 and 10 months. So, but it's now quite an efficient process to be able to take a protoplast and take it all the way through to a regenerated banana. That's the first time it's ever been done, as far as I'm aware, in Cavendish bananas, and we'll be certainly publishing this in the near future. Okay, so that was important. We then had to show that we could go and edit bananas and regenerate them to the point where there was no new DNA. So again, we used this PDS gene, and you'll see that we've got little on the left-hand side there, these little albino, these little white bananas regenerating. And so these were done without any selection whatsoever. We were able to confirm by what was edited using sequencing. And then very importantly, the 2 very, very important steps were to show by PCR that there was no new DNA that had been integrated. And finally, we sequenced the entire genome of 2 of those plants. That panel there is just demonstrating that from the entire genome, there was no new DNA that was being integrated into those plants. We found that this is actually quite efficient. With the PDS plants, we regenerated 31 plants. 9 of those 31 were edited in the way that we wanted them to be edited. And 8 out of the 9 were albinos, so they had all 3 genes knocked out. And so this is, and 2 of those plants have been completely sequenced, so we know they contain no new DNA. So we now have non-GM gene-edited bananas. So if this was a green one, and we do have equivalents of that, These bananas could be taken straight out of the lab. They could be planted anywhere because they're not regulated. These are non-GM, gene-edited Cavendish bananas, so a huge step forward for us. So then the next phase is the editing for Tr4 resistance. And that involves, firstly, identifying Cavendish genes that can be edited. You remember what I said earlier was that we needed to be able to alter genes already present in Cavendish bananas. So we're very lucky to have fabulous resources available to us. So we've got the complete sequence of Cavendish grand name, so we know all the genes there. We've actually assembled that genome. We've got the complete sequence of Malacensis 852. So that's the line that gave us RGA2. And then 848, which is also a Malacensis, but he's highly susceptible to both race 1 and race 4. That's a good comparator. But very importantly, we've got our QCAV4. So QCAV4 on the right-hand side there is identical to Cavendish grand name on the left-hand side, except for the presence of the RGA2 gene. So we've been able to go into that genome and find out what genes are upregulated and what genes are downregulated. That results in that resistance in QCAV4. So from that and other experiments, we now have identified a panel of 24 candidate genes that, whether they're upregulated or downregulated, have the potential to generate Tr4 resistance. So upregulation is in progress, upregulation and downregulation. So we've already put the edits for a number of these genes into Cavendish, and the most advanced of those are now regenerating. The next stage is taking those non-GM edited plants and challenging in the screenhouse. We plan to do that towards the end of 2022 up at our field trial site in the Northern Territory. And then from there, those that are identified there that having promising resistances in the screenhouse can go directly into the field. And as you saw from that photograph earlier, going from our screenhouse into our field is around about 50 metres. So it provides us with a fabulous opportunity to very, very rapidly screen for resistance in the screenhouse and then confirm that in the field. And at the same time as we confirm it in the field, the resistance, we can get all the data on cycle time, bunch weight, etc. So what are the progress and what are the opportunities? So we've now developed an efficient technology to generate non-GM gene-edited Cavendish bananas. We've identified a whole range of genes that have the potential to increase Tr4 resistance in Cavendish, and we're continuing with that. And we've also commenced editing Cavendish for Tr4 resistance, with those first ones probably going into the field by the end of next year or early 2023. So importantly with editing, as opposed to some of the other technologies, this allows us the continuous improvement of currently accepted banana cultivars. So Cavendish and Tr4 resistance is just the start. So we will hope to, probably within 2 years and maybe 3 years, have a Tr4 resistant Cavendish by gene editing. We can then go and edit that for other disease resistances or for other characteristics. So it offers us the opportunity to do a whole range of things. And that includes things like disease resistances, and not only Tr4 resistance, obviously. Black and yellow sigatoka are important, MOCO, etc. But also start to look at things like fruit quality, nutrition, and plant architecture. So all of these things are now becoming available because we can do the edits, and we can do them as non-GM edits, but also we have the genomes that we can go and look at and identify opportunities for the future. So it really is a terribly exciting place to be at the present time. Okay, I thought I'd finish with just a slide showing what countries around the world are now accepting gene editing in certain ways as non-genetically modified. And that includes most of North America and, amazingly, most of South America and Australia and Japan. The likelihood, those in yellow, and they're becoming more and more, the likelihood is that they're going to also accept non-GM gene editing, which is fabulous because that gets away from the concerns about genetic modification, be they only perceived concerns. The one problem we have at the moment is the European Union, which at the present time is saying genome editing is very is just a type of GMO and has to be regulated in the same way. That attitude is starting to change. It's starting to change in Britain, but there's a lot of discussion now in Europe about Europe missing out on these incredibly important technologies. So let me finish there. I'd just like to acknowledge we obviously have a very big team at QUT that is involved in this, and we've got fabulous funding support. So Fresh Del Monte is supporting us here, Australian Banana Research Company, and two Australian granting bodies are also supporting this research. So I would like to acknowledge those. So thank you very much for your attention. Sorry I was a bit late. That was obviously a technical glitch, but look forward to any questions.